folks, welcome back to Backyard Microscope. This is episode number 6. We're going to do something a little different today. We're going to talk about cacti. Now, if you're familiar with cacti, maybe you've brushed up against one, that's probably an encounter that you'll never forget. But there's some very interesting facts about cacti that we're going to look into. Some cacti can grow to 66 feet tall. Others are only a couple of inches tall. So there's a wide variety in the species there. They're well known for the absorption of water and water storage. And so they would be a very tasty meal to many organisms in the area. Tortoises, however, love to eat prickly pear cactus, and they're no worse for the wear having eaten those. But there's other features of these range of plants that are very interesting. They come in many colors: green, blue, brown, yellow, many different shapes and sizes. And as I mentioned, we'll talk about the water storage capability and the gel-like substance inside that we'll look at under several different types of microscopes. But when you study the literature about these things, it says something like they have spines instead of leaves. Now, leaves are completely different from spines, and we're going to show you how unique these spines are. These are not leaves, and they're not there to replace leaves. Actually, these things do stem photosynthesis. That's why they're green or blue or brown, because they're using a specific chlorophyll to do stem photosynthesis. And so they're very good at doing photosynthesis. They don't need leaves. In fact, leaves are a detriment in this dry, hot environment. Now, the spines do several things. First of all, they minimize the water vapor loss from the stoma, or the holes that are in the stem or leaf. In this case, those stoma are useful for exchanging gases with the atmosphere, one of which is water vapor. And so with all these spines arranged all around the body, it prevents excessive loss of water vapor, because it actually controls the flow of air around the body of the plant. The other obvious function of the spines is to ward off any animals that might otherwise consume these things and decimate them, because they're very easy to get to. They're low on the desert floor, and they can be eaten very easily. And so we're going to look at spines under the electron microscope. We'll talk about their water absorption capability, how they store this water. Other interesting facts are that their roots are very shallow, but they're very big. And so again, they're there to grab that water that percolates through the sand. So we hope you enjoyed this episode. So let's look at spines in detail. Cacti exhibit stunning diversity of forms and shapes. Here you see the saguaro cactus, which is a tall sentinel all over Arizona. The prickly pear, which is well known and grows wild even in California, Mexico. And so there are many different diverse forms. Here are barrel cacti, and we'll show you a couple of different pictures of these. But they all have the same basic features in that they have large roots that collect large amounts of water, and they store a lot of water within their stems. Now, they do not have leaves. Here is, we believe this is probably a form of Cholla, but not positive. We'll look at more Cholla here in a second, which is what we're going to concentrate on. Here now is Cholla, and you'll see several pictures of these. But they all have spines, and spines are not modified leaves, as the literature would lead us to believe. They seem to be designed to do specifically what they're designed for, which is to repel things that would damage or consume or eat or destroy these organisms, and to minimize water vapor loss. So we're going to concentrate in this episode on Cholla, jumping Cholla. You see here, these pieces break off on animal fur, or when you're walking through, they'll break off on your skin or on your long clothing. And so we're going to examine different features of this Cholla and show you things you've probably never seen before about cacti. And hopefully in the future, we'll also look at different types of cacti, maybe even the saguaro. And so, but one thing is characteristic are these spines. So let's take a look at these spines under the microscope. So what we'll do is we'll remove 2 or 3 spines from this piece of Cholla and examine them under a dissecting microscope, which at about 20x magnification. And the first thing you see here, the orange arrow is pointing to an actual spine, but these yellow arrows are pointing to sheaths or coverings that came off of those spines. And so this is a very interesting phenomenon. And in order to examine this, I want to show you video of these sheaths being pulled away. And here is one video, and I'll grab the spine with one needle nose forceps and I'll pull the sheath away with another. And so what is the purpose of these sheaths? Well, I think that these are a protective covering to protect the delicate nature of the spine that you will see in a little while when we magnify these spines under scanning electron microscopy. Here's another example. We'll hold the spine and pull the sheath off. You can see this delicate sheath, which is a sheer covering that lies over the spine to protect it. And we took some scanning electron micrographs of this, and here now is a picture of an SEM, and that arrow is pointing to the sheath. And we'll shoot another picture here in a second. Here are three white arrows pointing to the sheath or material or covering, and the gray arrow pointing to the actual spine itself. I want you to notice the detail of the spine with its longitudinal features, these columnar features of this hard spine itself. But notice that the sheath, this delicate gossamer thin material that covers over it, mirrors it exactly, so it's intimately associated with the surface of the spine to protect its delicate structures. Now here's a higher magnification image at the bottom of the spine where it's been cut away from the cactus, and remember the literature says that this is a modified leaf, but look at what the gray arrow is pointing to. It's pointing to the soft, sort of gel-like internal medulla in the inside of this spine, and there's no channels in here, there's no plumbing in here, there's no tubes to carry phloem and xylem, and so this is not a modified leaf. This is a spine designed to do exactly what it does. It's not a modified leaf. So we've shown you the internal soft medullary portion of this spine, but now look at what the white arrow is pointing to, and this is an outer hard woody exterior on this spine which gives it rigidity. If this were a modified leaf, it would not be covered with wood. Leaves are not covered with wood. They have plumbing inside in the midrib and the petiole, and they are stuffed with chloroplasts which do the photosynthesis. These spines do not do photosynthesis, and so this in no way is a modified leaf. It is a spine designed to do exactly what it does as a thorny spine. Furthermore, if this was a modified leaf, it would be covered in wax, and no spines are covered in wax. As we showed you before, these are covered in a thin gossamer sheath which protects the integrity of the spine. So spines are not a modified anything. They are designed to be spines, they function as spines, they are spines. Now I want to point out the nodes on the cholla, and these are encircled by these yellow ovals. And these nodes are the places where the spines and also the glockens, which are tinier spines, arise from the cactus. So we're going to show you spines, we're going to show you glockens, and we're also going to show you some very interesting structures, which I call tubes because I can't find them in the literature. Now here are some nodes that show up very nicely from a different cactus, and what I want you to see is all this fine material in the nodes. It looks like almost dust or glass or fibers of some kind. And these are another form of spine which are very tiny and very nasty called glockens. And so we'll have some better pictures of these to show you in a second. So here now are these glockens, these very nasty looking barbed sort of fibers, very thin. They're almost like shards of glass that embed themselves into your skin. The more you try to get them out, the more they work their way into your skin. So these are the nastier form of spines on the cholla called glockens. What I've done here is I've enhanced the image a little bit so you can see these tips. These are 4-micron tips, and that's about 1/100th the diameter of a typical hypodermic syringe that's used in the doctor's office. Maybe you noticed in one of the previous pictures these little tubes. They're off to the right on one of the previous pictures, but now here I've centered them up in this micrograph, And what are these tubes? These little tubes, I don't know if they connect down into the cholla and are used for gas exchange or what they are. I can't find them in the literature, so I just call them tubes. But also notice at the four corners of this picture, I've put an asterisk on each of the spines that is arising out of the node. So in the node, you have the four spines arising. You have the glockens, which are those nasty little tiny barbed spines. And then you have these tubes, for lack of a better term. I'll magnify them here just a tad more. And what are these things? They might be for gas exchange. It's really hard to tell because I can't find them described anywhere in the literature. So this might be new, never seen before. Here at long last now is one of the spines from the cholla. The tip of this spine is about 4 to 5 times smaller than a standard hypodermic needle that's used in a doctor's office. So these can penetrate your skin very easily and in multiple numbers. If you rub up against these cholla hard enough, you'll get many of these spines embedded in your skin. And notice now around the circumference all the way up and down the shaft of this spine are these little barbules that are pointed the opposite way from the tip. And so what happens is, as you try to pull these spines out of your skin, these little barbules open up, and you can see how small they are. They have 3-micron tips, very tiny. So they will open up and literally impregnate themselves in your skin in a way that you'll have to rip your skin away in order to get these out. So that's why these are so hard to get out of your skin once they've been embedded. Here is one barbule that's been separated out so you can see it. It's almost razor sharp with its 3-micron tip. So you definitely want to stay away from these kind of spines when you're walking around in the desert. So here's a poor girl who had a cholla embedded in her skin, and a friend with some very special gloves is trying to remove it from her skin, and you can see how hard it's pulling away the skin from her leg. And so when you do get these and you pull them out of your skin, make sure you disinfect with alcohol when you're done. One of the features that we mentioned about these plants that makes them perfectly suited for their hot, dry environment is their water retention capability. And here you see some of the wax that covers the outside of the plant. Here's another shot of the wax. And so these things actually manufacture a wax much like candle wax that they use to completely coat the outside of their stems in order to retain the water that they collect. So in this exercise with the jumping cholla, we're using a dissecting microscope, and I'm sorry I haven't taken more time to describe that to you. Essentially, it is a low power microscope, generally that you can zoom in and zoom out with. Here I have a TV adapter and a TV camera and monitor, so you can see what's going on under the microscope. And I also have these flexible fiber lights, flexible fiber optic illuminators. And that's very bright, but it's great because I can position this light anywhere I want it on my sample, and so it gives great shadowing and also great illumination for my sample. So we'll talk more about dissecting microscopes in a future episode and give you some tips on some of the best dissecting microscopes you can use. But as we've been discussing, we're talking about the jumping cholla, and here you see I've sectioned now a piece of it away from the rest of the body, and these are very nasty. These spines, as you've seen under the electron microscope, are pretty nasty, but we want to look at the internal structure to see if it autofluoresces and to see where the chloroplasts are. And so I have a piece of the green part of the cholla right here that I've sectioned away and cut off. And so now I'm going to work under the microscope to just take off a little section of this so that we can take some part of it and put it on a slide. So I'm just going to grab this as well as I can, and then with my scissors I'm going to try to attempt to just cut away a little piece that shows this gel-like structure inside. And it's a little tough to deal with because it's very gooey and sticky, so everything sticks to itself. But here now is a nice piece of the green body of the jumping Cholla, and it has a nice outer skin, and we'll show you better images of this shot through the microscope. But you can also see this internal very jelly material, and that's what we're going to put on a slide, this jelly stuff right here, and look at it under the fluorescent microscope and see if it autofluoresces. Here we're showing the outer dermis or skin of the Cholla, and you can see all these tiny little white holes. Those are openings or stomata for gas exchange. Okay, now here's a really cool experiment that you can do with any cactus, really. I have this little red cactus, red and green cactus here, and I tell you what's interesting. Why is this red? Well, it's red because it is using a red chlorophyll to do photosynthesis. So, as I've mentioned before, there are many different types of pigments for chlorophyll, but this is one we had on hand which has a nice open space on it, and there's a reason for that, because I'm going to do an impression of the surface here that you can look at later under the microscope. So what I did is I washed it with a powerful jet of distilled water. I washed down in that crevice there, and then I let it air dry, but I also blew it out with some canned air carefully. And now what I'm going to do is I'm going to paint these surfaces with some clear fingernail polish. And so I'm just going to try to put a couple of coats on here. And then, now I wouldn't recommend doing this over the whole surface of the plant because that would of course kill the plant. So I'm just going to put a few coats on here, and I'm going to let it dry, and then we're going to peel this off after it dries. And the peeling you can put on a microscope slide and look at it under the microscope. So we're going to stop the camera right now and let this dry, and then we'll come back and peel it off. Okay, so I think this is ready now. I think it's dry enough, but you have to be really delicate here, and you have to use a fine needle forceps to reach in here, and I'm just going to try to pull. Well, that's not very good. I want to pull away a large enough strip like that. You see that? I'm trying to pull this strip off, but then it keeps breaking. So it's a little bit of a challenge to pull this off in a strip that's long enough. This is looking pretty good. Well, let's see here. See if I can grab this. Oh, there's a nice strip. So I'm going to put that down on the microscope slide. I've got a little drop of water on there. Come on, you, stick down on that. Now it doesn't want to stick down. Well, the goal is to peel off a big enough strip to let it sit on that drop of water, and then let that drop of water dry up, and then you can look at this under your microscope. There's a nice little strip. See if I can get this to stick. These films are hydrophobic, which means they're afraid of water. So I think I'm going to have to go get another fine needle forceps in order to use two forceps to get this on the water. But you get the idea. Once you get that on the slide, then you can look at it under your microscope, and you'll see the surface of the cactus showing all of the stoma, all the openings in there that allow for gas exchange. So here are the results under a regular light microscope. It took me a few tries to get this film to point face up, but as you can see, the black lines are pointing to the open stomata, and the red lines are pointing to the cell boundaries, these really cool looking cells that have these squiggly lines. Here now a little higher power with the orange lines pointing to the cells, and the black ones pointing to the stomata, those open holes. And so this is kind of a fun project that you can do on your own it's going to take you a little practice to get it right, as it did me, but you'll end up with a really cool slide with the cell boundaries and the stomata showing up very nicely. Now the Cholla has this waxy outer skin that we looked at, and we saw the stoma in there, and now we're looking at this mucilage, this internal green goo. It's green because it does photosynthesis, and it also has other amazing properties. like amazing water storage and other things that are going to surprise you. So we'll talk about those in a second. But it's this green mucilage that we're going to look at under the fluorescent microscope in a little bit. Now let's use a still picture here to show you some of this anatomy. The orange arrow is pointing to that mucilage, that internal gooey green stuff that occupies most of the inside of the cholla. It's green because it does photosynthesis, and it's full of chloroplasts, which are the little engines that make the sugars in photosynthesis, and we're going to look at that under the fluorescent microscope. We'll show you those glowing chloroplasts in a second. But this green gooey stuff, this mucilage, is amazing. It has tremendous properties. It's like a gel that expands when it's placed in water, so it absorbs a tremendous amount of water. It's made up of polymers or chains of several different kinds of sugars, and so it's healthy to eat. In fact, studies show that it's kept sheep alive for 8 months living on that alone. It's non-toxic, and new studies actually show that this is a tremendous water purifier, even removing arsenic from water. So a nice bucket full of mucilage out in the desert draining your water is the ticket. And then we have the black arrow pointing to the dermis, which is the outer skin that's covered in wax and has those stoma that are sunken. They're sunken down in so that they will not lose much gas in exchange with the environment. So a tremendous amount of design in this unique plant. All right, so let's switch gears here a little bit, and we've taken a smear of this mucilage and we smeared it out on a slide and put it on a bright field microscope. And you see all those green structures. Those are all the chloroplasts that do the photosynthesis. But look at that rectangular structure. You remember the hollow tubes that we saw before? Well, that's one of those hollow tubes. And it's showing up fairly well in bright field, but let's go to fluorescence. And here we're using a UV fluorescence, and now you can see the tube and a lot of the mucilage glows, and so do these chloroplasts. The chloroplasts glow a bright red, and so we can see definitely that we have chloroplasts. Here's a little movie, and as you look through this, look at how much more mucilage you can see now under autofluorescence. Because remember, we're hitting these with a UV light, and they're giving off their own light. And then, of course, all the red chloroplasts glowing because they're full of chlorophyll, and the chlorophyll is being excited by the UV light. So once again, fluorescence microscopy and autofluorescence is really a cool thing. Let's do this again with blue light fluorescence, and here you see the chloroplast glowing red, and but the mucilage glows green instead of a blue. So in the blue light fluorescence, we get a different coloration. There's another tube that's glowing brightly. Now let's go back to bright field, and here's a tube, and it looks like it has some kind of material inside of it, and we're going to switch from bright field straight to fluorescence. Here's blue light fluorescence, and it's just eerie how this tube is glowing green and all the chloroplasts are glowing red. Here's another field of view. All of these circular structures are chloroplasts, and you'll see them glowing red here in a second, but some of them glow green, which means they have some lipids in them. And so epifluorescence, autofluorescence is a lot of fun to use with living plants.
Yuck. 9. This doesn't taste very good. I know you think it does. It's that goop from the cactus. Well, that is our episode. I don't know if you like it. I did not like it. Why tell the people about the spines? The spines are good. Spines stick to people, and then I laugh. Give me something to laugh about. But now for 8 months, I have to eat this goop. Why? All because I said I didn't hurt the baby in episode 1. I did not hurt this baby. This baby is fine. Anyway, I have to eat this now. That's our episode. Auf Wiedersehen.